this video, we are going to learn about the eukaryotic genome special features. So, just start with the main topic, special features of eukaryotic genome in detail. So, eukaryotic genomes have several unique features compared to prokaryotic genomes. Here are some of the most important features of eukaryotic genomes, like larger size. Eukaryotic genomes are generally much larger than prokaryotic genomes, while the smallest bacterial genomes are only a few 100,000 base pairs in length. Eukaryotic genomes can range from tens to millions to over a billion base pairs. Then introns. Eukaryotic genes often contain introns, which are non-coding regions of DNA that interrupt the coding sequence of a gene. Intron-containing genes are transcribed into RNA, but the introns are removed through a process called splicing before the final mRNA is formed. This allows for alternative splicing, which can generate multiple protein isoforms from a single gene. So, introns are eukaryotic genes contain introns, which are non-coding regions. Coding regions are non-coding regions. Non-coding regions of DNA that interrupt the coding sequence of a gene. Intron-containing genes are transcribed into RNA, but the introns are removed through the process called splicing before the final mRNA is formed. This allows for alternative splicing, which can generate multiple protein isoforms from a single gene. Multiple origins of replication. Eukaryotic chromosomes have multiple origins of replication, which are regions where DNA replication begins. This allows for efficient and rapid replication of the large eukaryotic genomes. Then, chromatin structure. Eukaryotic DNA is packaged into a complex of proteins called chromatin. The basic unit of chromatin is the nucleosome, which consists of DNA wrapped around histone proteins. Chromatin structure can regulate gene expression by making genes more or less accessible to transcription factors and other regulatory proteins. Next, telomeres. Eukaryotic chromosomes have specialized structures at their ends called telomeres, which protect the DNA from degradation and fusion with other chromosomes. Telomeres are composed of repetitive DNA sequences that are added to the ends of chromosomes by the enzyme telomerase. Then gene duplication. Eukaryotic genomes contain many duplicated genes which can arise through processes such as gene duplication and polyploidization. Then duplicated genes can evolve new functions or be retained as backups in case of mutation or environmental changes. Then non-coding RNAs. Eukaryotic genomes produce a variety of non-coding RNAs, which are RNA molecules that do not encode proteins. These include ribosomal RNA, transfer RNA, microRNA, and long non-coding RNAs, among others. Non-coding RNAs can regulate gene expression, participate in RNA splicing, and perform other functions. Overall, eukaryotic genomes are much more complex and diverse than prokaryotic genomes, reflecting the complexity and diversity of eukaryotic organisms themselves. Once again, let's have a quick recap of some other points related with this. We know that eukaryotic genomes are complex and diverse with many different types of sequences and structures. They have portions called introns, non-coding regions of DNA that interrupt the coding sequence of a gene and are removed during RNA splicing. Then exons, coding regions of DNA that are expressed as proteins. Promoter regions, regions of DNA that control the initiation of transcription. Then enhancers, DNA sequences that increase the rate of transcription. Silencers, DNA sequences that decrease the rate of transcription. Then transposable elements, DNA sequences that can move around the genome and cause mutations. Then some regions called telomeres, repeated sequences at the end of chromosomes that protect them from degradation. Then there are regions called centromeres, regions of DNA that attach chromosomes to spindle fibers during cell division. Then repetitive DNA sequences that are repeated many times in the genome, such as satellite DNA, mini satellites, and microsatellites. Then retrotransposons, transposable elements that move through an RNA intermediate. DNA transposon, transposable elements that move directly from one side to another in the DNA. Then histones, proteins that package DNA into chromatin. Nucleosomes, the basic unit of chromatin, consisting of DNA wrapped around histone proteins. Then epigenetic marks, chemical modifications of DNA or histones that can regulate gene expression. Then CPG islands, regions of DNA that are rich in CPG dinucleotides and are often found near the promoters of genes. Then CPG methylation. What is CPG islands? CPG islands, regions of DNA that are rich in CPG dinucleotides and are often found near the promoters of genes. CPG methylation, a common epigenetic modification in which a methyl group is added to the cytosine base in a CPG dinucleotide. Then heterochromatin region, a tightly packaged form of chromatin that is generally transcriptionally inactive. Heterochromatin is a transcriptionally inactive type, but it's highly condensed region. There are two types of heterochromatin, facultative and constitutive heterochromatin. And next, euchromatin, a more loosely packed form of chromatin that is generally transcriptionally active. Then alternative splicing, a mechanism by which different mRNA transcripts can be produced from a single gene, resulting in different protein isoforms. Then RNA editing, a process by which RNA sequences can be altered after transcription. Then non-coding RNAs, RNA molecules that do not encode proteins such as ribosomal RNAs, transfer RNAs and microRNAs. Then long non-coding RNAs, RNA molecules that are longer than 200 nucleotides and have regulatory functions. Circular RNAs, RNA molecules that form closed loops and can regulate gene expression. RNA interference, a mechanism by which small RNAs can regulate gene expression. Then alternative polyadenylation, a process by which mRNA transcripts can have different polyadenylate tail lengths affecting their stability and translation. Next, gene duplication, a process by which genes can be duplicated, leading to the evolution of new functions. Polyploidization, a process by which cells can have multiple copies of chromosomes, leading to increased genetic diversity. Then horizontal gene transfer, a process by which genes can be transferred between different species. Horizontal gene transfer, a process by which genes can be transferred between different species. That is horizontal gene transfer. Next, gene fusions, a process by which two or more genes can be combined into a single gene, leading to the evolution of new functions. Then genomic rearrangements, large-scale changes in the structure of a genome such as inversions, translocations and duplications. Eukaryotic genomes in detail, structure, function and importance. The eukaryotic genome is the entire set of genetic information that is carried by an organism's chromosome. It consists of DNA sequences that encode 4 genes, regulatory elements, repetitive elements and non-coding regions. The structure, functions and importance of eukaryotic genome are discussed below along with the examination techniques used to study it. Structure of eukaryotic genome. Eukaryotic genomes are housed within the nucleus of cells and are much larger and more complex than prokaryotic genomes. They are organized into linear chromosomes, with each chromosome containing many genes and regulatory elements. They are organized into linear chromosomes, with each chromosome containing many genes and regulatory elements. The DNA is tightly packaged around histone proteins to form nucleosomes, which are then further compacted into higher order structures. The arrangement of chromatin into chromatin and heterochromatin regions is also important in regulating gene expression. Next, learn about the functions of eukaryotic genome. The eukaryotic genome is responsible for encoding all of the genetic information necessary for an organism's growth, development and survival. It does this through a variety of functions including: next, encoding proteins. The primary function of genes is to encode the information related to synthesized proteins. These proteins can perform a wide range of functions, from catalyzing chemical reactions to structure support and movement. Then regulating gene expression. The genome also contains a wide range of regulatory elements that control when and where genes are expressed. This regulation is critical for development and differentiation and for responding to changes in the environment. Maintaining chromosome structure. The genome is responsible for maintaining the structure and integrity of the chromosomes. First point was that the first function is encoding of genetic information necessary for organisms' growth, development, and survival. Then encoding proteins. These proteins perform a wide range of functions from catalyzing chemical reactions to structural support and movement. Then regulating gene expression. The genome also contains a wide range of regulatory elements that control when and where genes are expressed. This regulation is critical for development and differentiation and for responding to changes in the environment. Then maintaining chromosome structure. This also helps in maintaining chromosome structure. The genome is responsible for maintaining the structure and integrity of the chromosomes. This is critical for proper segregation during cell division and for maintaining genetic stability over generations. Next, importance of eukaryotic genome. The eukaryotic genome is critical for the survival and function of organisms. Understanding the structure and function of genome is essential for many areas of biology, including evolutionary biology. Comparing the genomes of different organisms can provide insights into the evolutionary relationships between them. It's also used in medical research, like understanding the genome is critical for studying genetic diseases and developing treatments for them. Then biotechnology, genetic engineering, and other biotechnological applications rely on the ability to manipulate and understand the genome. Then examination techniques. There are several techniques that are used to examine the structure and function of a eukaryotic genome, including DNA sequencing. This technique allows for the determination of the order of nucleotides in a DNA molecule, providing information about the structure and function of the genome. Then chromosome staining. Like different staining techniques can be used to visualize the chromosomes and identify structural abnormalities. Then fluorescence in situ hybridization or FISH. FISH can be used to detect specific DNA sequences within the genome, allowing for the identification of specific genes or chromosomal regions. Then chromatin immunoprecipitation chip technique. Chromatin immunoprecipitation technique. Chip technique is used to identify protein DNA interactions within the genome, providing information about regulatory regions and chromatin structure. Then CRISPR-Cas9 genome editing. CRISPR-Cas9 allows for the precise multiplication of genome, allowing for the identification of gene function and the development of new biotechnologies. In summary, the eukaryotic genome is a complex and dynamic structure that is critical for the survival and functioning of organisms. Understanding its structure and function is essential for many areas of biology, and a range of techniques are used to study it. Special features of eukaryotic genome. Once again, we are going to learn that. So, the eukaryotic genome is the complete set of genetic information carried by an organism's chromosomes. Unlike prokaryotic genomes, eukaryotic genomes are much larger and more complex. They are organized into linear chromosomes and are tightly packaged around histone proteins. The eukaryotic genome is responsible for encoding all of the genetic information necessary for an organism's growth, development, and survival. Here are some of the special features of the eukaryotic genome, like introns and exons. One of the most notable features of the eukaryotic genome is the presence of introns and exons. Introns are non-coding regions of DNA that interrupt coding sequences called exons. The process of splicing removes introns from the pre-mRNA transcript, allowing exons to be joined together to form the final mRNA molecule. This process greatly expands the complexity of the eukaryotic genome, allowing multiple proteins to be produced from a single gene. For example, the human gene that encodes for protein dystrophin contains 79 exons, and mutations in this gene can lead to the development of muscular dystrophy. Then non-coding DNA. Another feature of the eukaryotic gene is the presence of large amounts of non-coding DNA. Non-coding DNA makes up the majority of human genome and its functions are still being studied. However, it is known that non-coding DNA can contain regulatory elements that control gene expression as well as repetitive sequences that have been implicated in genome instability and disease. For example, trinucleotide repeat expansions in the non-coding regions have been linked to diseases such as Huntington's disease and Fragile X syndrome. Then chromatin structure. Now chromatin structure, the organization of chromatin is also a significant feature of the eukaryotic genome. The DNA is tightly packaged around histone proteins to form nucleosomes, which are then further compacted into higher order structures. The arrangement of chromatin into euchromatin and heterochromatin regions is important for regulating gene expression. Euchromatin is generally less compact and is associated with active gene expression, while heterochromatin is more compact and is associated with gene silencing. For example, the inactivation of one X chromosome in female mammals is accomplished by the formation of heterochromatin on one of the X chromosomes. Then transposable elements. Transposable elements, also known as jumping genes, are sequences of DNA that can move from one location in the genome to another. They can disrupt gene function and cause mutations, but they can also contribute to genetic diversity and evolution. Transposable elements are found in all eukaryotic genomes and some make up a significant portion of the genome. For example, in maize, transposable elements make up to 85% of the genome. Gene duplication. Gene duplication is another important feature of the eukaryotic genome. It occurs when a gene is duplicated, resulting in multiple copies of the same gene. These copies can then undergo mutations and diverge in function, leading to the development of new genes with different functions. Gene duplication has been implicated in the evolution of complex organisms as well as the development of novel traits. For example, the human genome contains multiple copies of the gene for alpha globin, which is important for the production of hemoglobin. In conclusion, the eukaryotic genome is the complex and dynamic structure with many unique features. These features allow for the production of multiple proteins from a single gene, regulation of gene expression, genome stability, genetic diversity, and evolution. Techniques such as DNA sequencing, chromatin immunoprecipitation, and CRISPR-Cas9 genome editing are used to study the eukaryotic genome. Alternative splicing. Alternative splicing is a process by which exons can be selectively included or excluded from the final mRNA transcript, allowing for the production of multiple protein isoforms from a single gene. This process greatly expands the diversity of the proteome and estimated to occur in up to 95% of human genome. What is the alternative splicing process? Alternative splicing is a process by which exons can be selectively included. Exons can be selectively included or excluded from the final mRNA transcript, allowing for the production of multiple protein isoforms from a single gene. This process already expands the diversity of the proteome and is estimated to occur in up to 95% of human genes. For example, the genes for the protein tropomyosin can produce over 40 different isoforms through alternative splicing. So that is a special feature of eukaryotic genome, alternative splicing. Then epigenetic modifications. Epigenetic modifications are changes to the DNA and histone proteins that do not involve changes to the underlying DNA sequence. These modifications can affect gene expression and are often heritable and one cell division to the next. Epigenetic modification. Epigenetic modifications are changes to the DNA and histone proteins that do not involve changes to the underlying DNA sequence. Epigenetic modifications. Epigenetic modifications are changes to the DNA and histone proteins that do not involve changes to the underlying DNA sequence. These modifications can affect gene expression and are often heritable from one cell division to the next. Some common epigenetic modifications include DNA methylation, histone acetylation, and histone methylation. Epigenetic modifications are important for regulating gene expression during development and can be influenced by environmental factors. Some common epigenetic modifications include DNA methylation, histone acetylation, and histone methylation. Epigenetic modifications are important for regulating gene expression during development and can be influenced by environmental factors. For example, exposure to high levels of stress during early development can lead to changes in DNA methylation that increase the risk of mental illness later in life. So, exposure of high levels of stress during early development can lead to changes in DNA methylation that increase the risk of mental illness later in life. Then, genome rearrangement. Eukaryotic genomes are subject to frequent rearrangements such as translocations, inversions, and duplications. These rearrangements can affect gene function and can have been linked to the development of genetic disorders such as cancer. However, they can also contribute to genetic diversity and evolution. For example, the rearrangement of genes involved in the development of immune system is thought to have played a key role in the evolution of vertebrates. So next point is genome rearrangement. Earlier we have learned about alternative splicing, then epigenetic modifications, then like methylation and all such points. Then genome arrangements. Eukaryotic genomes are subject to frequent rearrangements such as translocations, inversions, and duplications. These rearrangements can affect gene function and have been linked to the development of genetic disorders such as cancer. However, they can also contribute to genetic diversity and evolution. For example, the rearrangement of genes involved in the development of immune system is thought to have played a key role in the evolution of vertebrates. Then non-coding RNAs. In addition to coding for proteins, the eukaryotic genome also encodes for a variety of non-coding RNAs or ncRNAs. This include transfer RNAs, tRNAs, and ribosomal RNAs, rRNAs, involved in protein synthesis, as well as regulatory RNAs such as microRNAs, miRNAs, and long non-coding RNAs. These ncRNAs play important roles in regulating gene expression and are implicated in a wide range of diseases. For example, miRNAs have been linked to cancer, while incRNAs are involved in the development of brain and nerve system. Gene regulation networks. Finally, the eukaryotic genome is organized into complex networks of gene regulation that involve multiple signaling pathways, transcription factors, and regulatory elements. These networks allow for precise control over gene expression in response to environmental cues and developmental signals. Disruptions in these networks can lead to developmental abnormalities and diseases. For example, mutations in genes involved in the regulation of sonic hedgehog signaling pathway can lead to developmental disorders such as holoprosencephaly. Chromatin remodeling. Chromatin remodeling refers to the process by which the packaging of DNA into chromatin is altered to allow for changes in gene expression. This can involve changes in the positioning of nucleosomes and modification of histone proteins and the recruitment of transcription factors. Chromatin remodeling is important for regulating gene expression during development and in response to environmental cues. For example, changes in chromatin structure have been linked to the development of cancer. Then alternative polyadenylation. Alternative polyadenylation is a process by which different polyadenylated tails are added to the 3' end of mRNA transcripts, resulting in different lengths of the 3' translated region, UTR. This can affect mRNA stability and translation efficiency. Alternative polyadenylation is thought to play a role in gene regulation and has been implicated in a variety of diseases including cancer. Then RNA editing. RNA editing is a process by which the nucleotide sequence of RNA transcripts is altered after transcription. This can involve the conversion of adenosine to eosine or the insertion or deletion of nucleotides. RNA editing can change the amino acid sequence of the protein encoded by the mRNA transcript or affect its stability or function. RNA editing is important for regulating gene expression and has been implicated in a variety of diseases including neurological disorders. Then transposable elements. Transposable elements are sequences of DNA that can move from one location in the genome to another. They can be classified as either retrotransposons or DNA transposons. Transposable elements can disrupt gene function and contribute to genome instability, but they can also play a role in evolution by introducing new genetic variation. For example, transposable elements are thought to have played a role in evolution of the immune system in vertebrates. Next, gene duplication and divergence. Gene duplication and divergence is a process by which a gene is duplicated and one of the copies evolves to perform a new function. This process can lead to the evolution of new genes and the expansion of gene families. Gene duplication and divergence is important for evolution of new functions and has been implicated in the development of a variety of structures and symptoms, including the vertebrate and the mammalian brain. Then telomeres. Telomeres are specialized DNA sequences that cap the ends of chromosomes and protect them from degradation and fusion with other chromosomes. Telomeres are essential for maintaining chromosome stability and preventing the loss of genetic information during cell division. Telomere shortening has been implicated in aging and age-related diseases, while telomere lengthening has been linked to cancer. Next, RNA interference. RNA interference is a process by which the expression of specific genes is inhibited by small RNA molecules such as miRNA and siRNA. RNA is important for regulating the gene expression during development and in response to environmental cues. It is also used as a tool for gene knockdown in research and has potential applications in the development of therapeutics. Then genome size variation. Eukaryotic genomes vary greatly in size, ranging from less than 10 MB to over 100 GB. Genomic size is not always correlated with organizational complexity, with some simpler organisms having larger genomes than more complex organisms. Genome size variation is thought to be influenced by a variety of factors, including transposable elements activity and polyploidization. Horizontal gene transfer. Horizontal gene transfer is a transfer of genetic material from one organism to another outside of the normal process of reproduction. Next, horizontal gene transfer or GT is the transfer of genetic material from one organism to another outside of the normal process of reproduction. HGT can occur between organisms of the same or different species and can lead to the acquisition of new functions or traits. HGT is thought to have played a role in the evolution of prokaryotes and may also occur in eukaryotes, although its frequency and significance in eukaryotic evolution is still under debate. Then genome editing. Genome editing is a technology that allows for the precise modification of the genome at specific locations. This can involve the insertion, deletion, or replacement of genetic material. Genome editing has potential applications in medicine, agriculture, and basic research. The CRISPR-Cas system is a popular genome editing tool that has revolutionized this field of genetics. Alternative splicing. Alternative splicing is a process by which different combinations of exons within a pre-mRNA transcript are spliced together to generate multiple mRNA isoforms. This can lead to the production of different protein isoforms from a single gene and is an important mechanism for regulating gene expression. Alternative splicing is thought to be involved in a variety of biological processes including development, cell signaling, and diseases. Then epigenetic modifications. Epigenetic modifications are changes in the DNA or histone proteins that affect gene expression through changing the underlying nucleotide sequence. These modifications include DNA methylation, histone modification, and chromatin remodeling. Epigenetic modifications are important for regulating gene expression during development and in response to environmental cues. It can also be heritable, meaning that it can be passed from one generation to the next. Then non-coding RNAs. Non-coding RNAs are RNA molecules that do not encode proteins but have a variety of other functions in the cell. These functions include regulating gene expression, chromatin structure, and translation. Examples of ncRNAs include miRNA, incRNAs, and snRNAs. These are thought to have important roles in development, disease, and evolution. Alternative splicing coupled to nonsense mediated decay. Alternative splicing coupled to nonsense mediated decay is a mechanism for regulating gene expression that involves the degradation of mRNA transcripts that contain premature stop codons. This mechanism is used to prevent the translation of potentially harmful truncated proteins. ASNMD is thought to be involved in regulating gene expression during development and in response to environmental cues. Then chromosomal aberrations. Chromosomal aberrations are alterations to the structure of a number of chromosomes that can lead to the genetic disorders and cancer. Examples of chromosomal aberrations include deletions, duplications, inversions, and translocations. Chromosomal aberrations can alter gene expression and disrupt normal cellular processes leading to disease. Then transcription factors. Transcription factors are proteins that bind to specific DNA sequence and regulate gene expression. They play important role in development, cell differentiation, and disease. Examples of transcription factors include homeobox which are involved in patterning during development, and oncogenes, which promote cancer cell growth and proliferation. Then genome duplication. Whole genome duplication is a process by which an organism's entire genome is duplicated. This can lead to an increase in genetic diversity and the evolution of new functions. Genome duplication events have occurred multiple times in the history of eukaryotes and are thought to have played a role in the evolution of vertebrates and angiosperms. Then gene regulatory networks. Gene regulatory networks are networks of genes and their regulatory elements that interact to control gene expression. These networks are important for regulating gene expression during development and in response to environmental cues. Gene regulatory networks are complex and dynamic and involve feedback loops and other regulatory mechanisms. Next, comparative genomics. Comparative genomics is the study of similarities and differences in genome structure and function between different organisms. Next, comparative genomics is important for understanding evolutionary relationships between organisms and for identifying genes and pathways that are important for specific biological process. Then, personalized genomics. Personalized genomics is the use of genomic information to tailor medical treatment and preventative measures to an individual's genetic makeup. This approach has the potential to improve the efficiency and safety of medical interventions and reduce healthcare costs. Personalized genomics is an emerging field that is still in its early stages. The eukaryotic genome plays a critical role in the structure, function, and regulation of eukaryotic cells. Importance of eukaryotic genome include the basis of inheritance. The eukaryotic genome contains all the genetic information necessary for the development, growth, and function of an organism. It is passed down from generation to generation and is the basis of inheritance. The eukaryotic genome is involved in a variety of cellular processes including DNA replication, transcription, and translation. These processes are essential for the proper functioning of cell and rely on the accurate transmission and interpretation of genetic information. Then gene regulation. The eukaryotic genome contains regulatory elements that control the expression of genes. Gene expression can be regulated by a variety of mechanisms including transcription factors, epigenetic modification, and non-coding RNAs. Then there are developmental processes. The eukaryotic genome plays a critical role in developmental process such as cell differentiation and morphogenesis. These processes are controlled by the expression of specific genes and are essential for the proper development of tissues and organs. Then evolutionary process. The eukaryotic genome is subject to evolutionary process such as mutation, recombination, and selection. This process contributes to genetic variation within and between species and can lead to the evolution of new traits and functions. Then disease process. The eukaryotic genome is involved in the development of many diseases including genetic disorders, cancers, and infectious diseases. Understanding the genetic basis of this disease is essential for developing effective treatment and preventative measures. Then biotechnology. The eukaryotic genome has important applications in biotechnology including genetic engineering and gene therapy. These technologies rely on the ability to manipulate and modify the eukaryotic genome to achieve specific outcomes. Then conservation. The eukaryotic genome contains important information about the evolution and diversity of life on Earth. Understanding the structure and function of the eukaryotic genome is essential for conserving biodiversity and protecting endangered species. Then personalized medicine. The eukaryotic genome is increasingly being used in personalized medicine to tailor medical treatments and preventative measures to an individual's genetic makeup. This approach has the potential to improve the efficiency and safety of medical interventions and to reduce healthcare costs. Then fundamental research. The eukaryotic genome is the subject of expensive research in genetics, genomics, and other biological sciences. Understanding the structure, function, and regulation of the eukaryotic genome is essential for advancing our understanding of basic biological process and for developing new technologies and applications. Then epigenetic inheritance. The eukaryotic genome is subject to epigenetic modifications like DNA methylation and histone modification that can be inherited across generations. These modifications can influence gene expression and have important implications for development, disease, and evolution. Then genome stability. The eukaryotic genome contains mechanisms for maintaining genome stability, such as DNA repair pathways and checkpoints during cell cycle. These mechanisms help to prevent mutations and maintain the integrity of the genome. Then non-coding DNA, which we have just mentioned earlier. Then chromosome structure, which we have already learned. Then comparative genomics is also learned. Then omics technologies. Recent advances in genomic, transcriptomic, proteomic, and metabolomic technologies have revolutionized our ability to study the eukaryotic genome and its functions. These technologies allow researchers to study the genome at multiple levels of organization and to generate large amount of data for analysis. Then bioinformatics, the analysis and interpretation of genomic data require sophisticated computational tools and methods. Bioinformatics is a rapidly growing field that is essential for the analysis and interpretation of genomic data and for the development of new technologies and applications. Then ethical considerations. The use of eukaryotic genome in research and medical applications raises important ethical considerations such as issues of privacy, consent, and equity. These issues must be carefully considered and addressed to ensure that the benefits of genomic research and applications are realized in an ethical and reasonable manner. Interdisciplinary collaboration. Studying the eukaryotic genome requires collaboration across multiple disciplines, including genetics, molecular biology, biochemistry, biophysics, computer science, and ethics. Interdisciplinary collaboration is essential for advancing our understanding of eukaryotic genome for the development of new technology and applications. Public engagement. The eukaryotic genome has important implications for society, and public engagement and education are essential for ensuring that the benefits of genomic research and applications are widely understood and accepted. Effective communication and engagement of the public are critical for building trust and support for genomic research and applications. Thank you for listening.